Hello, everyone. Welcome to today's live broadcast, a streamlined approach for reversed phase method development using a combined column screening and software modeling approach. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this web seminar presented by LCGC and sponsored by MacMod. A company should be able to condense its mission statement into a simple phrase. MacMod's mission statement is smarter chromatography. But you may ask, just what does smarter chromatography mean? It means that when they make a recommendation to you about an HPLC or UHPLC product, it's to partner with you to offer innovative solutions to your complex problems. They use advanced state-of-the-art analytical software to match their product portfolio with your separation needs. They leverage their 30-plus years of experience in the LC column business, their technical expertise, and their manufacturing network to help you solve your toughest application problems. They provide up-to-date and accurate technical catalogs and reports from industry-leading separation scientists to keep you informed of new technologies. Before we get started today, we have a few housekeeping announcements. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, which you can find on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. And if you have any technical problems viewing or hearing the presentation today, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speaker. We are very pleased to be joined today by Alan McEwen, the Business Development Director at Advanced Chromatography Technologies Limited. Alan McEwen has more than 20 years of experience in method development and separation science technology innovation in the pharmaceutical industry with AstraZeneca and Pfizer, and in LC column R&D with Advanced Chromatography Technologies. He has published various articles and has written several book chapters on separation science, chromatography, and genotoxic impurities. Alan is a Fellow of the Royal Society of Chemistry and a Chartered Chemist. Alan, thank you for joining us today. Please get us started.
Okay, thank you very much for the introduction, and welcome to everybody this afternoon to this webinar, Streamlined Reverse Phase Method Development using a Combined Column Screening on Software Modeling Approach. So my name is Alan, and my email address is on the slide there, should you want to send questions directly to me rather than the Q&A session with this webinar. We'll also be sending out a copy of these slides as a PDF after the event. So moving on with the show, this webinar is split into three sections. So we have, first of all, a brief discussion on method development and strategies. Then I'll talk a little bit about selectivity, the key to chromatography, and how systematic screening of certain parameters can be very useful for your method development approach. And then I'll move on to talking about software and how a particular type of software that we've had some success with, ChromSort software, can be used for more complex separations and how modeling can help you in your method development activities. So normally method development is the exciting part of chromatography. This is the part of the LC work that we do which always is interesting. We have a problem in front of us and we can't wait to get our first column onto the instrument and begin making some injections to see if we can resolve the compounds we're interested in. Now, of course, for most people, method development is, as I say on the slide, an iterative process. We change some parameters, we make some injections, we look at the influence that that change has had on the retention and separation of our compounds, and we make a judgment on what to do next. Now, there are different ways to do method development. There's the trial and error approach, as I've just explained. Some people do a stepwise approach to method development, a so-called one factor at a time. So systematically, they work through different chromatographic parameters and see how that influences the retention and separation of the compounds they see in their samples. There are other approaches as well, the so-called quality by design approach to method development. This is something that is had more influence in different industries over the last 10 to 15 years or so to try and fully understand and risk assess different parameters and how they can affect the separation of critical components in your samples and on your chromatogram. And then there's systematic screening and its use for method development. Now, from my perspective, I'm a big fan of systematic screening because any well-structured approach to method development is only going to be helpful. It's also something that generates data in a logical fashion and allows you to understand how the different parameters are affecting your components in your sample. Different types of parameters in chromatography have different impacts on retention, separation, and resolution of the compounds. Selectivity is an integral part of that process. Also, if you're doing screening of different parameters, you can semi-automate this depending on the instrumentation that you have, whether you're using different types of instrument vendors. Normally there is an option to use different solvent systems or different column systems, and of course program your methods to use different types of parameters and parts of those parameters. Now, of course, the screening process combines these different parameters, and you program these into your sequence to generate data, which you then review after the sequence has completed. So, which are the important parameters for us to focus on in our method development? Well, on the slide now we can see the resolution equation, one form of the resolution equation. And this is from sound chromatographic theory, and it's very helpful to us in method development. We can see from the equation there's an efficiency component, a selectivity component, and a retention component. But of course, looking at the equation on the slide there, it doesn't help us understand which part of that equation contributes most to the resolution. And of course, we're trying to resolve analytes from each other, which is why this equation is important. When we look at this equation in a graph format, we can see now resolution on the y-axis, and on the x-axis we can see alpha, selectivity, n, efficiency, the number of plates, and k, retention factor. We can see that selectivity really is quite powerful with respect to resolution. In fact, it's the most powerful term in that equation. So selectivity really is the key to resolution, and of course parameters such as efficiency boost the performance of chromatography. So now we know that selectivity is the most powerful term in the resolution equation. Which factors affect selectivity in chromatography? From a practical perspective, in fact, many of the different parameters that we can change and modify have an influence on selectivity. And these are in a list format on the slide. So for instance, on the left-hand side of the slide under isocratic separations, we can see we have column stationary phase type, we have pH and organic modifier right at the top of that list as being very influential parameters on selectivity. On the right-hand side of the slide, we have gradient separations, gradient dilution chromatography, very common nowadays because many of the samples we receive have different hydrophobicities, different lipophilicities, and so we need to change the percentage organic in our mobile phase to elute everything in a reasonable time frame. For gradient chromatography, the parameters on the left, of course, are just as important as for isocratic analysis, but we have some additional parameters specific for gradient chromatography. Gradient steepness, we have K-star, which is the apparent retention in gradient chromatography from the equation, the shorthand equation I've listed on the screen there, which is gradient time, the flow rate, the gradient range, column volume, and a constant depending upon the analyte molecular weight. We also have dwell volume and also column dimensions. All of these parameters can affect selectivity to a greater or lesser degree. And of course, when we're doing method development, it makes sense to focus on those parameters that give us more impact on selectivity. But this is going to help us develop our method more effectively. In this talk, I'm going to talk about column stationary phase type, or more accurately, the different types of mechanisms of interaction and how they can help us maximize selectivity. I'm also going to talk a little bit about organic modifier type and how we can combine column stationary phase and the organic modifier type into a systematic screening protocol that maximizes selectivity and is very useful for method development, particularly when it's set up on an instrument for automated screening. When we're designing new stationary phases, we want to consider those different mechanisms of interaction. And we can see on the slide here that we have a simple ring system. And if we substitute onto this ring system different types of electron donating groups, and I've put some examples on the screen there. We've got some amines, some alkylamines, alkoxy type groups, other types of aromatics, then we change the properties of that ring system. You can see, for instance, I've drawn a simple alkyl-phenol type stationary phase. We can see that there is a weak dipole moment across that group, and the ring is slightly electron-rich or electronegative. Now, of course, we can do the opposite to that when we're modifying from a chemistry perspective these different types of functional groups, and we can add, for instance, electron withdrawing groups. And again, I've got some examples on the slide there. And I've drawn the classic pentafluorophenol stationary phase. So this is a very popular stationary phase that is known to give a different and very useful type of selectivity for certain compound types and classes in method development. Now, the pentafluorophenol is actually an electron-deficient ring system. We've got those 5 big fluorine groups substituted onto the ring system, which create a moderate dipole across that ring. Now, of course, from a chemistry perspective, this is all very interesting, but what does it mean for chromatography? Because that's what we do, and how can we use this in method development? Well, you can see on the slide there that what we've effectively done is combine the properties and robust chemistry of a C18, but together with the aromatic ringer of the ring system to create the C18AR, for instance, on the left-hand side of the slide. And on the right-hand side of the slide, we've got the chemistry of the C18, but together with the pentafluorophenol stationary phase. We've effectively combined all of the best parts of a C18 chemistry and a phenol chemistry, or a C18 chemistry and a pentafluorophenol chemistry. These stationary phases have multiple mechanisms of interaction. This helps us to maximize selectivity. Rather than just have hydrophobic interactions, which is the dominant mechanism for a C18 chemistry, we have other types of mechanisms present too. This helps us maximize alpha or selectivity, therefore we maximize the resolution of our compounds. These stationary phases also have useful other elements too. For instance, they have a low bleed profile. Because we have an extended alkyl chain connecting to the aromatic groups, it means that the hydrolysis of the ligands is low. They have the same bleed profile as a standard C18. So this is very useful for gradient LC-UV work and also excellent for LC-MS type analysis too. And finally, these type of stationary phases from rational phase design, they're also 100% wettable. So that means that you can use 0% organic solvent in your mobile phase. For instance, when you're trying to retain polar compounds and you want to drop the organic as low as possible, with these phases you can drop the organic solvent content to 0, 100% aqueous mobile phase, to try and retain those very polar hydrophilic species on your chromatogram so you can integrate them and quantify them. So these are a good example of rational phase design. They maximize selectivity with other positive parameters too. And we can demonstrate these type of mechanisms and the differences in selectivity with some simple chromatography. So here we can see this is a mixture of regioisomers or positional isomers. So I'll show the structures at the end. And we perform an isocratic analysis, and there appears to be 4 peaks on this chromatogram, maybe 4 compounds. This is with a standard C18 column, and of course the only mechanism of interaction, the dominant mechanism, is hydrophobic interactions. Now with the C18AR column, we introduce the aromatic group. So we introduce pi system interactions as well as hydrophobic interactions, and you can see we have a different selectivity, and we appear to have seen or found another compound. So there are now 5 peaks in this chromatogram. But finally, if we use the C18PFP with the additional mechanisms of the moderate dipole and also this shape and positional selectivity given by that stationary phase chemistry, we can see there's actually 7 components in that particular mixture. And I'll put the structures of the regioisomers on the slide there as well. So you can see how the different chemistries really do give different chromatography and different selectivity. So stationary phase selectivity is very powerful for method development. Now there's other types of stationary phase that we've designed as well. For instance, here we have a polar embedded phase, which many different vendors offer polar embedded phases. But we can see here that between the polar embedded group, the embedded amide, and the silica particle, we have an extended alkyl chain spacer. So this gives us more stability to the stationary phase. And then we also have a stable cyanochemistry too. And you can see here again an extended alkyl chain spacer, which reduces the hydrolytic instability of this type of phase. But we still get the mechanisms of interaction of that terminal nitrile. So all of these phases give a very different selectivity to each other. Different selectivity, hopefully maximizing the resolution in our method development. These phases as well give different mechanisms of interaction, low bleed profiles, so they're excellent for our CMS work, and are also 100% wettable, so you can retain polar compounds. And here's a nice example which shows under exactly the same conditions a separation of catechol and resorcinols. So these are polyhydroxyaromatic compounds of moderate polarity. And we can see under exactly the same conditions on a C18 column, on the chromatogram at the bottom, the compounds are bunched up close to the T0 with low K values on the chromatogram. With the C18 amide, under exactly the same conditions with exactly the same sample, we can see a very different profile. So that embedded amide group providing different mechanisms of interaction to give us different retention and selectivity. So how do we understand the different selectivity, the different mechanisms on these different column chemistries? Well, we use something what's called the Tanaka characterization protocol, which I've cited there at the bottom of the slide. We use this approach for designing our stationary phases and also ensuring we can understand the mechanisms of interaction. And on the slide now you can see 6 different column chemistries, and we can see 5 different mechanisms of interaction: hydrophobic binding, pi system interactions, dipole-dipole interactions, hydrogen bonding, and shape selectivity. The Tanaka protocol is a very simple protocol that that you run certain compounds down each of the different column chemistries, which are defined in the protocol under certain conditions. You calculate ratios from the retention factors of different compounds, and this gives you a value. And the size of this value indicates the weighting of different mechanisms. So for instance, with a standard C18 column, we perform the different tests from the Tanaka protocol, and you can see that unsurprisingly, hydrophobicity is a dominant mechanism with standard C18 columns. You can see that there is no pi-pi system interactions because there's no aromaticity to a standard C18 stationary phase. There's no dipole-dipole interactions because there's no polarizable functionality with a standard C18 phase. There's a small amount of hydrogen bonding, and this is from the silanols that are present on the silica surface. And also, maybe surprisingly for some people, you can see that many C18 columns have a small degree of shape selectivity, and this is because of the densely bonded C18 chains and the way that analytes can move into the C18 chains, and you can get a degree of shape selectivity depending on your analytes. Now, if we look at the rest of the different mechanisms, you can see there's different weightings of the different types of mechanisms. So there's different pi system interactions, dipole-dipole interactions, hydrogen bonding, and shape selectivity. So of course, from a method development perspective, it makes sense to use different column chemistries that have different mechanisms to try and tease apart the analytes in our samples to give us the resolution that we're interested in. It makes no sense to do method development with multiple columns of the same type because you have the same mechanism of interaction. If you have different mechanisms, it can help you resolve your compounds. And that's the approach that we take in this systematic screening approach. So we have different stationary phases with different mechanisms, and they're all complementary selectivity to each other. 6 columns together in a screening platform. Now the approach that we actually use in my lab, and we use this every day, is we have actually 6 different column chemistries, but we also use the power of solvent selectivity too. If you remember, that was third down on the list of parameters that influence selectivity. So you can see that we have in blue methanol, the protic solvent methanol. It has a different selectivity effect, different allotropic strength compared to in red acetonitrile, the aprotic solvent acetonitrile. And so we use the solvent selectivity together in combination with the chemistry and mechanisms of interaction of the different columns to give us our screening platform. So this is a 6-column, 2-solvent method development platform. This is what we use for all of our method development screening. So this is a picture of the instrument that we use in my lab, and you can see here we have a UHPLC instrument. It has a column manager next to the instrument, and we have a switching valve, and you can see the 6 different column chemistries placed inside the thermostatic oven. Now the standard format that we use for our screening is 100 millimeters by 3 millimeters, 2 micron particle size. This allows, well, what we found is this is a good compromise of speed and robustness and column lifetime. So this allows us to have a reasonable number of plates for good, efficient chromatography. And also with the 2 micron particle, it means we don't have too high a back pressure, which means that the system itself is a pretty robust system. Now the example that I'm going to show you is based on our standard approach. So here's an overview of the method development in general. So step 1, we perform some scouting runs with general starting conditions using a combination of the 6 columns and 2 solvents. Then if we don't get an option for separation from that first systematic screen, we move into the optimization of the separation or for peak shape and runtime and so on. Then we can validate the method, and then we can implement the method for sample analysis. How do we calculate our starting conditions? Well, typically we perform a broad scouting gradient run with our samples to begin with, normally under acidic conditions. It's always under acidic conditions unless the application indicates otherwise due to sample instability or some other physicochemical parameter which would indicate acidic pH would not be suitable. How do we calculate the actual conditions for starting? Well, we solve this simple equation that I showed you earlier. For K values, retention values greater than 2 and less than 20, and we know the other parameters in the equation. So we know the gradient range, or the gradient range, we know the column volume and the constant, and then we can adjust TG, the gradient time, and the flow rate because we have a UHPLC instrument, and so we can adjust these 2 values to give us solve the equation for 2 and 20. Now the example I'm going to show you to begin with, the first example, is based on acetaminophen or paracetamol as it is in Europe. This is the principal component, the parent compound in this sample. We have 9 other impurities or degradants that are spiked into the sample at low levels. And our goal is to separate all of these components from each other. So this is a typical related substances type method development activity. Excuse me. Now, if we have physicochemical data available to us, it's always useful to review the data. Now, because acetaminophen is a very well-known compound, it's been around for 40 years plus now, we have the structures available to us, We have ionization constants, we have lipophilicity data, log D values, the distribution of different forms at different pHs. All of these different data are available to us. Now, whenever we start a method development activity, and particularly for this one where we have known compounds, it's important to assess the data to make sure the starting conditions are appropriate. When we look at the data for these different compounds, something becomes very clear to us. And we can see there for 4-aminophenol, it has a negative log P value. So log P is a measure of lipophilicity. A negative log P value means that this compound is very hydrophilic. In fact, it's so hydrophilic, it's so polar, that it is not retained under acidic conditions in reverse phase chromatography. So of course that's a problem. All compounds need to be retained so we can integrate them and quantify them in our samples. If we look at the log D values though, as we increase the pH, you can see the polarity of this compound goes from a negative value to a positive value. So this indicates that our starting conditions, instead of being acidic pH, actually mid pH could be a better separating area, a better area to retain this compound. And so for this work, we used pH 6. Most of our screening work is done at pH 3 using ammonium formate and formic acid mobile phase. But for this work, we used ammonium acetate at pH 6 so we could retain the 4-aminophenol. So we prepare the sample, we make it up, we filter it, we put it on the instrument, we set the sequence running, and at the end of the day, we have 2 sheets of paper on our printer next to the instrument. There is 1 sheet of paper with 6 chromatograms in methanol. Each of the 6 chromatograms corresponds to a different column chemistry. And then a second sheet of paper, 6 chromatograms and 1 for each column chemistry in acetonitrile. That's our standard first step to our method development. So this is the actual data from the instrument. So we can see if we start at the top there, we have the columns going down to the bottom, the C18, C18AR, the C18PFP, the super C18, the C18 amide, and the cyano-ES. First thing to note is compound 2 on every chromatogram here is the 4-aminophenol, the most polar compound in this mixture. And you can see that even at pH 6, the K value is low, but it is retained away from the T0 suitably enough so that we can integrate it and quantify it. If we look now at some of the chromatography, so the green boxes, you can see the parent compound, acetaminophen, peak 1, and you can see an impurity, peak 4, co-eluting with the parent on the C18. And you can see as we move down the chromatograms with the AR, it's on the tail, co-eluting with the PFP, co-eluting with the super. On the C18 amide, the polar embedded phase, compound 4 elutes before the parent compound. So quite a different selectivity compared to the others so far. And then with the cyano-ES, the stable cyano phase, compound 4 elutes after the parent. So different mechanisms of interaction, different retention and elution profile. If we look at peak 7 and 8, they're very well separated on the C18, the AR, the PFP and the super C18. But when we come down to the polar embedded phase of C18 amide, you can see that they've actually switched around in elution order. And they're still reversed on the cyano-ES. So those 2 columns giving us quite a different interaction compared to the other 4 columns. And then finally, peaks 9 and 10, starting at the top, we have co-elution, complete co-elution. We have just about separated, if you zoom in on the PFP, co-elution with the super C18, well separated with the amide, and well separated with the cyano-ES. And if we count up the number of peaks that are resolved across the 6 chemistries in methanol, don't forget this is the first solvent, methanol, you can see we've actually got 2 situations where from the standard screen with no further method development, we have complete separation of all 10 components in this mixture, which is fantastic. So we've set up the sequence, we've walked away and let the instrument and the chemistries do the work for us, and we have 2 options where we have all 10 compounds separated. So that shows you how a screen can be useful. Perhaps the most important point from this slide, however, is this. We can see here the red box is around the C18 chromatogram because that's where 95% of people would start their method development using a C18 column because it's the most common column used all around the world in all different application areas. And we can see that the C18 would not have provided separation. In fact, you can see we have compounds eluting early in the chromatogram, peaks 1 and 4, and then we have compounds eluting later in the chromatogram, peaks 9 and 10. Then we would need to do further method optimization. Whereas if we perform the screen as I've shown, we have two options at the bottom, the polar embedded and the stable cyano, where we have complete separation. So that's the first 6 chromatograms in methanol. What about acetonitrile? So again, we can look at peak 2, and we can see here it is retained on the chromatogram. The K value is low, retention is low, but it is retained, so we can integrate it and quantify it. Now, if we look at the chromatography, peaks 1 and 4, this time working from the top down again, we can see separated on the first 2 columns, begins on the PFP to elute close to the tail, well separated on the super C18, co-eluting with the C18 amide, and well separated with the stable cyano phase. Now, with peaks 7 and 8 and 9 and 10, peaks 8 and 9 begin to interfere. So we can see working from the top with the C18, we have co-elution, we have some separation with the AR, separation with the PFP, separation with the super C18, separation with the C18 amide, and also separation with the stable cyano phase. And if we look at the number of peaks resolved again, we can see this time we have 4 options for separation. And again, if we look at the C18, we have an area of co-elution. So those people who started with the C18 would require further method development. So the point I'm trying to make from these 2 slides is that the screening approach is semi-automated. It's based around selectivity using the different column chemistries together with the 2 different solvents, the protic methanol, the aprotic acetonitrile, and it's provided us with options. That's useful and a good start to method development. However, of course, it doesn't always work like this, because otherwise I would be out of a job. So it doesn't always work like this because sometimes we don't get any options for a method, and so we need to do further optimization work. Now, the approach that I've shown you just now, the systematic screening, is actually available as a free A2 poster. We've got a very nice poster you can use on your lab wall. If you'd like to email me or email someone at MatMod, then they will be able to send you that poster to stick on your lab wall. So how do we optimize the separation when it doesn't work? So we're looking now, once we've done the screening, at optimization. The column screening allows us to choose the best combination of column and organic solvents. So now we need to take a step further and optimize other parameters. And of course, there's lots of other parameters that we could optimize, and I've listed some on the screen here. Gradient time, the percentage B range of the solvents. We can look at temperature, we can look at pH, we can look at buffer concentration. There's many other different types of parameters. Now, method development software for more complex separations can be very useful. And what I want to talk a little bit now is a package called ChromSort. So this is a method development software that you can use to generate models to help you predict and also optimize your separations. We've used it extensively in reverse phase chromatography, and it can be used for a number of different parameters, but it can also be used in other types of modes of chromatography too. Now, what you can do with this software, and I'm going to show you a simple optimization of the gradient time using a gradient retention model. You can input from as little as 2 initial runs, although typically we use 3. And then you can start to do a simple modeling of the gradient to see if you can improve your separation. So you input the 3 different input runs, gradient, the run time, the gradient time with retention, and you can simply generate your first model. And this will then simulate the separation in the software. And there's other different types of software packages out there that do very similar things as well. So on the bottom left of the screen, you can see the grid. This is on the y-axis, this is the percentage organic, and on the x-axis, you can see there's time. That's the gradient profile, and then you can see the simulated chromatogram and the right-hand side of each of those different panels. And you can see it, for instance, on the right, where I've adjusted the gradient down, the green line, and you can then see how this affects the simulated chromatogram. So that you can do manually, but also the software can optimize for you. So there's an element of optimization that it will perform on your behalf based on the input data that you've put into the software. So for instance, you can see that on the right-hand side now, we've got the gradient profile again, the green line, but it has suggested a gradient profile to give us separation of the compounds based on the data that we've inputted. And you can see the simulated chromatogram on the right. So I'm going to look at a more complex example now and how the software can help us. So here we have our standard screening approach that I've already explained. Now this sample is more complicated. This is a triple API example. So this has 3 different components in it, plus it has 14 other compounds, impurities and degradants, making a total of 17 analytes in this sample mixture. The 3 different compounds are present at higher proportions compared to the impurities. So again, this is simulating a related substances type situation. Now you can see the screening data on each of the 6 different column chemistries, and this is in methanol, as you can see clearly from methanol in the top right hand corner of the slide. I've ringed on the chromatograms, and they're in the same order as I've done previously, the C18, AR, PFB, super C18, the polar embedded C18 amide, and the cyano-ES. I've ringed with red rings the different areas of coelution. And you can see there's multiple areas of coelution on all of the different column chemistries in methanol. If we look at if we look at the number of peaks resolved, you can see that we have a maximum of 11 compounds out of the 17 resolved. So again, this is still not obviously perfect. We want to separate and be able to integrate and quantify all 17 components. Using the standard screen and methanol, there are 11 components maximum resolved. If we look at acetonitrile, the same screening conditions, the same column chemistries, but using the aprotic solvent acetonitrile, we can still see coelution on all column chemistries. So this is a nice example compared to the acetaminophen example I showed you earlier, where we had lots of different options where all components were resolved from the screen. In this example, we don't have any options where all the components are resolved from the screen. And again, we can see the maximum number of peaks resolved is 11 on the C18 amide. So from our flow diagram, we need to choose the best combination of column chemistry and solvent and take this forward into our optimization phase. For this particular example, we use the polar embedded C18 amide column with methanol. So we have the 5-minute screening gradient inputted into the software. We also inputted the 10 and 15-minute gradients to be able to generate our model. And then we could use the software to optimize and propose to us and the optimum linear gradient, and we also did optimization for the optimum step gradient as well to see what the software would give us. But when we inputted those 3 simple runs based on obviously very simple injections on our instrument, collecting the retention data and inputting them into the software, you can see the optimum predicted linear gradient. So this is a predicted chromatogram from the software, and you can see the gradient profile that was proposed by the software, which should give us that chromatogram. So this was the predicted data, and of course it's always important to experimentally verify, and we can see the experimental data based on the real samples. And of course you can see that first of all, the error between the predicted retention times and the experimental retention times were pretty low. So the model was quite good. However, when we look, and I've tried to highlight with numbers 1, 2, 3, and 4, we can see there's some areas where we would perhaps prefer there to be better resolution. And so even though the software thinks in the predicted chromatogram that we have full separation, of course software is based on optimum interactions, optimum peak shape, and so when we get to the real life experimental data, sometimes there may be some tailing or there may be other things going on which don't give us exactly the same as the predicted model. So we can see there that we would ideally like to further resolve the peaks, peak pairs highlighted in 1, 2, 3, and 4. So I'll hold that thought for the moment. So what about step gradients? What does the software give us in terms of options for step gradients? So we can see here we have a target runtime of 10 minutes, and we allow the software to predict what it thinks we should do for a 10-minute runtime to separate all 17 components. So here we have the predicted chromatogram on the top, and then at the bottom you can see we have the experimental chromatogram. And again, you can see, I highlighted again some peak pairs 1, 2, and 3. There's suboptimal resolution of these compounds because the software has predicted perfectly Gaussian peaks and how it can resolve all of those compounds, but in reality, due to load and tailing and various other effects, you can see that experimentally we still need to better resolve some of those compounds. So the question is, for instance, in this example, can we move the peak 5 away from peak 4 to improve the resolution? Now with the ChromeSword software, we can identify a peak pair as important, so we can increase the weighting to resolve that peak pair most importantly in the chromatogram. So it's almost like choosing your critical pair in the chromatogram, and the software will try to optimize those first and foremost and better than it has done previously. So when we allow the software to do the optimization, you can see this is the predicted chromatogram on the left-hand side. There is more resolution between peaks 4 and peak 5. And in fact, we can manually edit the gradients as well, even though it's predicted the gradient. You can see here that we basically ramped up the gradient to make sure we brought everything off the column. So we predicted the gradient, then we manually edited as well. And you can see here the predicted separation on the top and the experimental separation on the bottom, and the resolution values calculated for the peaks pairs identified as 1, 2, and 3. And we can see there that we have the final separation, which was successful. All 17 compounds are resolved from each other using a simple gradient time modeling approach. So hopefully what you've heard today is that selectivity is really important in chromatography. Systematic screening is a really important approach that I'm a big fan of. It's logical and allows you to semi-automate the system so you can go away and do other things. I've identified stationary phase chemistry, or more importantly, the mechanisms of interaction, and 2 organic modifiers, acetonitrile and methanol, which are very popular in chromatography, and using those together in combination to explore the selectivity of a separation in the screening approach. And what I've presented is a 6-column 2-solvent approach for method development, and I demonstrated a 10-compound example using acetaminophen and how the screening approach gives us options based straight from the screen. And then I went on to explain a more complex example where the screen didn't give us full separation, and where we then used some software to help us develop a model which then could optimize further for us, and then we could prove the separation based from the conditions offered by the model. The software ChromeSword 2 we found to be useful and helpful in this type of modeling approach, and obviously today I've just shown a very simple gradient time modeling. Other options are available within the software. This is the product itself. You can see it's an A4 type size, and there are 6 different column chemistries in this product, and in the center you have a USB stick with getting started and all the manuals, and then a dongle for the software as well. So that brings me to the end of my presentation. I hope you found that useful. I will very happily try and answer some questions, but first of all, I will pass back over to the guys at LCGC so they can do some other housekeeping before we move on to the Q&A. Thank you very much.
Thank you very much, Alan, for that informative presentation. We do indeed have some things to do before we get started with the Q&A, and quite simply that's asking everyone in the audience to participate in 2 brief polling questions. And the first one is already on your screen. Please click directly on the screen to enter your answers. So here is that first question. Would you be interested in a personal consultation on your method development of your method utilizing the new Chrome Sword software? Yes or no? So once again, you can click directly on your screen to enter your answer. And that question is, would you be interested in a personal consultation on your method development for your method utilizing the new Chrome Sword software? Yes or no? Okay, excellent. So let's move to our second polling question. And that question is, see it on my screen now, from which manufacturer do you currently source your columns? Waters, Agilent, Phenomenex, Toso Bioscience, Merck Millipore, Thermo Fisher Scientific, or MacMod? So once again, click directly on your screen to enter your answer. Which manufacturer do you currently source your columns from? Waters, Agilent, Phenomenex, Toso Bioscience, Merck Millipore, Thermo Fisher Scientific, MacMod. And that list is long, so if the list is getting cut off, you might need to enlarge your slide window. Okay. Fantastic. Well, let's move on. It's now time for the question and answer period with the audience. I just want to remind everybody that if you'd like to submit a question, you can do that by typing it in the Q&A box, which you can find on the right-hand side of your window. All right, so let me pull up the first question. So here's one for you, Alan. Can this approach be used for both small molecules and biomolecules or biologics?
Hello. For large molecules, selectivity is not as a column chemistry and its effect on selectivity is not as powerful as it is for small molecules. You can certainly use software for modeling retention and creating retention models with larger molecules, but the column chemistry type approach, the wide range of column chemistries and how they influence selectivity is not the same for large molecules as it is for small molecules. For larger molecules, the column chemistries have a smaller impact, and the differences in column chemistry have a smaller impact on alpha. So I'd say that this approach is particularly useful for smaller molecules. There will be a place for larger molecules, but I don't think it's quite as powerful. For peptides and those type of molecules, this approach is useful and can be used, but where that cutoff is, I'm not sure. That's probably not something I'm really experienced with at the moment. I hope that answers the question.
How does Chrome Sword compare to DryLab?
So I've used many different types of software. I've used Chrome Sword, DryLab, LC Simulator, Fusion, and bespoke software as well. So many of these packages are broadly similar to each other. Their interfaces are different, and some of them have slightly different features and bells and whistles, if you like. So I guess, you know, I wouldn't really want to say one was particularly better than the other. We've used Chrome Sword very successfully. We've combined Chrome Sword together with our products because we found it to be pretty intuitive, but I wouldn't want to put anyone off any other packages or other types of options that are out there.
Is this software being used for teaching purposes?
I don't know if it is being used for teaching purposes. I can't see why it couldn't be used for teaching purposes. There's no reason why it shouldn't, you know, why the background theory of how it predicts retention, the relationship between different parameters and retention, all of that is based on sound chromatographic theory. There's no reason why it shouldn't be, but I'm not aware of any examples where it is.
What are the other capabilities of the software besides adding step gradients?
So the software can be used with multiple different parameters. I think I only showed one parameter today. Obviously, we've only got time to demonstrate one element of the software, but it allows you to predict retention, optimize retention, generate different models, and use that as part of your structured chromatography and method development approach. We've used it very successfully in our lab.
Are the columns available in HPLC and UHPLC column formats?
Yes, they are. Yes, the columns that I discussed today, the 6 different chemistries, are available in 1.7 micron, 2, 3, and 5 micron, and multiple different IDs from 2.1 to 4.6, and column lengths as well. So it doesn't matter whether you're doing UHPLC method development or HPLC method development, we will have one of those options available with this particular product. So it can be used across the whole different range from LC to UHPLC.
Can you confirm again what's included with the ACE ChromeSword product?
So the product itself is 6 different column chemistries of the same dimensions. So all of the columns will be, for instance, 154.63 micron or 50 by 2.1, 1.7 micron. There'll be 6 different column chemistries in there: the C18, C18AR, the PFP, C18amide, the Cyanide-S, and the Super C18. So the different mechanisms of interaction that I discussed. There's also a USB stick in the product as well, which has all of the information, the getting started, the user manual and so on for the software, and also the catalogs and further information on the different column chemistries, example applications and so on. And then the last thing that's in there is the actual dongle, which you need to plug into your laptop or your PC to activate the software. And that then allows you to begin the modeling process. And I should say as well, it wasn't on the slides, is for this particular package, it's priced at a very competitive point. And because of that reason, what we've done is the software is a limited time software. It's a 6-month license, but there are options to either upgrade or you can buy another product and get a further 6 months. So there's different options available there for you, but because of that, we've been able to bring the price point to a very competitive point in terms of modeling software, because normally modeling software can be quite expensive, particularly when you have other modules added on and so on.
Would you suggest acetonitrile or methanol as the eluent for a very unknown non-polar herbicide?
I would probably suggest, well, I mean, again, really, using the approach I've discussed, of course, I would say you should screen both and use 6 column chemistries, because that's what I've just said. In reality, if that sample was put in front of me, I would screen it on 6 chemistries and 2 solvents, because that's what we use in our lab. It's the first approach we use for everything. If you really wanted to push me and say, really, what would you use? Which one would you use? Because we can't use 2 solvents for whatever reason. Depends how non-polar it is. If you're saying it's quite non-polar, then obviously acetonitrile is a stronger solvent. So if you're concerned it may stick to the column, then maybe acetonitrile is a better option. But it would depend on your column chemistry as well.
Can you easily validate a method with step gradients?
Can you easily? Well, that's quite a general question. I have done in the past myself. I mean, nowadays with the instrumentation that we have, the quality of the engineering from multiple different vendors, the production of the gradients and typically the electronic feedback mechanisms they have for flow control and so on normally means the gradients are pretty good. So if you program in a certain gradient profile, it's unusual for it not to deliver that gradient profile as long as you're not really abusing your systems. So nowadays, it shouldn't be a problem. I know that the best approach for many methods is a simple isocratic method, obviously, because it's this, if you're keeping it as simple as possible, it's easier to execute, it's easier to validate, and so on. So when you compare it against that, a step gradient is a little bit more complex. But nowadays, with the instrument engineering, there really shouldn't be a problem to validate step gradients. And I've done that myself in the past without any problems. But of course, there's always going to be examples where people have struggled for whatever reason.
How do you choose the right pH solvent?
So our standard approach is to use acidic conditions. And in fact, if you look at most methods used, particularly in the pharma industry, but other industries as well, most methods are acidic in nature, so they usually have some sort of acid modifier in there, whether it's a formate or a citrate or whatever, a phosphate. Most modifiers or additives are acidic in nature. And there's a second reason for that as well, because many of the reverse phase separations, and reverse phase is the majority of methods that are developed in a few industries. Many of the methods, the columns are based on silica, and silica is more stable under conditions between 2 and 7. So acidic pH is sort of naturally chosen. But as I mentioned in my talk, there are example times where you can't use acidic conditions because of instability or some other physical chemical reason that your compound doesn't retain, changes form, degrades, or whatever. So for the first example, as I explained, at low pH for the 4-aminophenol, the compound is not retained under acidic conditions, and you can see that quite clearly. I didn't show the chromatogram, but when you run the chromatogram at pH 3 with that sample, you don't see the retention 4-aminophenol. It elutes on the T0, which is obviously not ideal because it means that you're not able to integrate and quantify that compound. So our standard conditions are pH 3, ammonium formate, unless we know otherwise. And for that first example, I use pH 6 because as we increase the pH, we get more retention of the 4-aminophenol because it's less polar. And in the second example, I used our standard screening conditions, which was ammonium formate at pH 3. And when people get the slides, they'll be able to look at the detail there. But you can see that it was done using our standard screen.
Here's another one. I would think pH screening is much more powerful than organic solvents. What do you think?
So it can be, of course, assuming that your compounds are ionizable. If you've got multiple neutral species in there, then changing the pH is going to have a minimal effect on your on the retention and separation of your compounds. pH overall is probably the most powerful parameter. The problem with screening different pHs is that oftentimes the preparation of the different pH media is done incorrectly. So you're not comparing like with like. Particularly when you go across a wide pH range, you will need to change buffer salts. Otherwise, you don't have a true buffer where it opposes small changes in pH. If you think of acidic conditions, we're looking at formates or citrates or phosphates. Mid pH conditions, it could be phosphates or acetates, and maybe bicarbonates as you go higher, right up to high pH where you have ammonium hydroxides and other types of additives. So you've got very different types of buffer salts. So if you're comparing low pH with mid pH and high pH, it's very difficult to do a true pH comparison. Particularly if you want it to be LCMS friendly, because you have to change buffer salts. So you have the impact of the buffer salt change on your retention and separation and selectivity, and you have to take that into account. And then the way that you make those buffers, making them as true buffers as opposed to adding acid and adding base to change the pH, that's also important as well. If you add acid and add base, you'll change the ionic strength as opposed to the buffer concentration of your mobile phase. Ionic strength also affects selectivity, separation, and so on. So you're changing multiple parameters. So that's why pH can be very powerful, but it's also got to be used in a very careful way.
Excellent. Well, with that, I think we should wrap up because we're about out of time. Alan, thank you so much for your talk today and for answering all those questions.
No problem at all. Thank you very much. Thank you for the questions.
I also want to thank everyone in the audience for attending and participating in today's event. And I'd like to thank our sponsor, MacMod, for making today's webcast possible. You will receive an email from LCGC alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.